Welcome. This presentation examines the molecular immunology of ovarian cancer, with emphasis on the immune pathways and therapeutic targets that are shaping modern immunotherapy strategies. Scientific presentation, prepared by Brian Geniuson, research scientist. Ovarian cancer, particularly high-grade serous ovarian carcinoma, often abbreviated HGSOC, represents one of the most complex immune environments among solid tumors. Unlike melanoma or certain lung cancers, ovarian tumors are frequently described as immunologically cold. This means that the tumor microenvironment often contains relatively low levels of activated cytotoxic T cells, alongside multiple mechanisms that suppress immune activation. Within this microenvironment, tumor-associated macrophages commonly polarize toward M2 phenotypes, promoting tumor growth and inhibiting effective immune responses. Myeloid-derived suppressor cells further limit T-cell activation, while stromal signaling pathways such as transforming growth factor beta contribute to immune exclusion and reduced immune cell infiltration. Together, these mechanisms allow ovarian tumors to evade immune surveillance. A central regulatory system controlling immune responses in cancer is the immune checkpoint network. Two of the most important checkpoints involved in ovarian cancer immunotherapy are the PD-1, PD-L1 pathway, and CTLA-4 signaling. The PD-1 receptor is expressed on activated T cells. When PD-1 binds to PD-L1 on tumor cells or immune cells within the tumor microenvironment, an inhibitory signal is transmitted that suppresses T-cell activity. This interaction can lead to T-cell exhaustion, reducing the ability of immune cells to recognize and destroy tumor cells. Checkpoint inhibitor therapies block these inhibitory signals and help restore immune activity against cancer cells. Another important biological dimension in ovarian cancer involves genomic instability and DNA repair defects. Many ovarian tumors exhibit homologous recombination deficiency, frequently associated with mutations in the BRCA1 or BRCA2 genes. These defects impair DNA repair and lead to the accumulation of DNA damage. Fragments of damaged DNA may enter the cytoplasm of tumor cells, activating an innate immune-sensing pathway known as cGAS-STING. Activation of this pathway promotes type 1 interferon signaling and enhances immune recognition of tumor cells. This molecular relationship helps explain why therapies that increase DNA damage, such as PARP inhibitors, may enhance immunotherapy responses in selected tumors. In addition to PD-1 and CTLA-4, researchers are investigating several emerging immune checkpoints that may contribute to immune resistance. Examples include LAG3, TIM3, and TIGIT. These receptors regulate immune exhaustion through mechanisms distinct from PD-1 and may represent important targets for next-generation immunotherapy strategies. Another promising therapeutic approach involves targeting tumor-specific surface antigens. In ovarian cancer, one important example is folate receptor alpha, commonly abbreviated FR-alpha. FR-alpha is frequently overexpressed on ovarian tumor cells and has become a target for antibody-drug conjugates and other precision oncology approaches. Targeting this receptor allows therapies to selectively deliver cytotoxic agents to tumor cells while minimizing damage to surrounding healthy tissue. Because ovarian tumors often employ multiple layers of immune suppression, modern research is increasingly focused on combination therapy strategies. These strategies may combine immune checkpoint inhibitors, PARP inhibitors, anti-angiogenic therapies, and cellular immunotherapies such as CAR-T approaches. The goal of these combined approaches is to increase tumor immunogenicity, reduce immune suppression within the tumor microenvironment, and enhance immune cell infiltration into tumor tissue. In summary, ovarian cancer immunotherapy is shaped by a dynamic interaction between tumor genetics, immune checkpoints, and the tumor microenvironment. Understanding these interconnected molecular systems is essential for developing more effective precision oncology strategies and improving therapeutic outcomes in ovarian cancer. Scientific presentation, prepared by Brian Geniuson, research scientist. This material is intended for scientific and educational discussion.